Hello everyone, welcome back to the class. In our previous class, we learned about role of SCDK in initiating DNA replication in S phase. So the pre-replicative complex plays an important role which assembles in replication origin, marking the preparation for DNA replication in G1. SCDK, that is S-phase cyclin dependent kinase, leads to the formation of multi-protein pre-initiative complex. Pre-initiative complex unwinds the DNA at the origin and begins the process of DNA replication. The key player here is ORC, origin of recognition complex, or origin recognition complex. Origin recognition complex is the large multi-protein complex. It binds to the origin in early G1. CDC6, CDT1 acts as a co-activators for ORC and forms a protein complex. This protein complex assembles with MCM protein, that is mini chromosome maintenance, which acts as a helicase. It is a hexamer of 6 related polypeptide, that is MCM227, and forms a ring structure. It acts as a licensing factor which allows replication to initiate. SCDK activation in late G1 phase triggers assembly of several additional protein complex at the origin, leads to the formation of pre-initiation complex which unwinds the helix and begins the DNA synthesis. As it initiates DNA replication, SCDK also triggers the disassembly of some pre-replicative complex components at the origin. By this, it blocks the re-replication by triggering the destruction of CDC6. If CDC6 is not activated, or if CDC6 is not binding with the ORC, then ORC will be inactive. So that ensures each origin is activated only once in each cell cycle. So this was the summary table of major cell cycle regulatory proteins and its function. Starting from CDK activating kinase to SCF, we have discussed each and every regulatory proteins. In today's class, we will be learning about cell cycle checkpoints. Cell cycle checkpoints plays a crucial role in seeing or in determining the perfect cell cycle procedure. So it functions to ensure the complete perfect chromosomal replication and passing to the daughter cell. So it is highly regulated cell cycle. It makes sure the highly regulated cell cycle. It ensures fidelity. It ensures the proper attachment of chromosomes. It ensures and guarantees from the prophase to anaphase. Each and every process should be happening in a particular time and period, in a particular procedure pathway. So this cell cycle checkpoints checks for each and every component of a cell division. So let's dig into this concept. So to begin with, let us quote some importance of cell cycle checkpoint and who hypothesized this concept. See, this cell cycle checkpoints functions to ensure that incomplete or damaged chromosomes are not replicated and passed on to the daughter cells. In most cells, there are several checkpoints in cell cycle at which cycle can be arrested if previous events have not yet been completed. So the cell cycle is highly regulated and a dependent series of events which are mediated by a number of checkpoints. So these checkpoints, or the cell cycle checkpoint concept, was hypothesized by two major persons, Hartwell and Weinert. Hartwell and Weinert hypothesized the existence of checkpoint pathway, by which they said that uncompleted cell cycle will send inhibitory signals. This inhibitory signals halt the later events. event. So Hartwell and Wiener hypothesized the existence of checkpoint pathways, by which they hypothesized that uncompleted cell cycle event sends an inhibitory signals to the later events. In most cells there are several checkpoints. So majorly to classify the cell cycle checkpoints, we take three major checkpoints. They are cell cycle checkpoint, second one is DNA damage checkpoint, and the last one is spindle assembly checkpoint. So cell cycle checkpoint, DNA damage checkpoint, spindle assembly checkpoints are the three major checkpoints in the cell cycle, which ensures the complete perfect without defect cell cycle. So let us know about them one by one. First we'll take the cell cycle checkpoint. What does this do? So this cell cycle checkpoint ensures the fidelity of the cell cycle. Ensures fidelity of cell division. These checkpoints verify whether processes at each phase of the cell cycle have been accurately completed. So it makes sure for the process to accurately completed. Accurately completed. Next, and it also ensures the phase of the cell cycle should be accurately completed before progression into the next phase. An important checkpoint in G1 has been identified in both yeast and mammalian cell. In yeast, this point is known as start, and in mammalian cell it is known as restriction point. Restriction point at which the cell becomes committed to DNA replication and also completes the cell cycle. It is during this G1 phase itself the cell integrates two type of signals, that is mitogenic and next one is growth inhibitory signals. If there is any defect at the G1 phase, this checkpoint will integrate the growth inhibitory signals. If everything is okay, then it integrates mitogenic signals, making the decision to proceed or pause or exit the cell cycle. So cell cycle checkpoint ensures the fidelity of the cell division. Next, the second one is spindle assembly checkpoint. Spindle assembly checkpoint. So this spindle assembly checkpoint is also known as mitotic checkpoint. It guarantees that anaphase onset is initiated only when all chromosomes are properly attached. So it makes sure for proper attachment of chromosome, especially metaphasic chromosome on the metaphase plate, and it monitors the correct attachment. It monitors the correct attachment of chromosomes to the mitotic spindle. Chromosomes to mitotic spindle. And also it prevents the segregation. It prevents the segregation of chromatids until they are properly aligned on the metaphase plate. So it prevents segregation of sister chromatids until they are properly aligned on the metaphasic plate. So let's discuss about each of these function of the spindle assembly checkpoint. So first, during mitosis, chromosome segregation is carefully monitored to prevent aneuploidy. So the spindle assembly checkpoint is majorly important to prevent this aneuploidy. So to do so, there are some mechanism when microtubules from two opposite spindle poles attached to the kinetochore of a metaphasic chromosome, a tension develops. So tension develops across the paired kinetochore owing to the mitotic force that tend to pull the sister chromatids toward two opposite poles. It is at this stage there are some defects that can occur. So the spindle assembly checkpoint makes sure there is nil defects. So it makes sure it prevents the defects at this stage. How? So there are some correction mechanism. So I'll write a diagram. You can just get a glance of it. So kinetochore is a protein complex, okay. So there are error correction mechanism which are done by kinetochores. So that is mediated by Aurora B kinase, which adds phosphate group to either serine or threonine residue. So you have come across this Aurora B kinase before, where it was during the discussion of role of mCDK during mitosis, where condensin protein was phosphorylated systematically by cyclin CDK, Polo and Aurora B kinase, right? This mCDK influenced the assembly of mitotic spindle by phosphorylating proteins that regulate the microtubule behavior. That was the point where we studied the Aurora B kinase, right? So it is the point where with the help of the Aurora B kinase, the kinetochore eliminates erroneous or incorrect microtubule attachment with Aurora B kinase, kinetochore harbors an error correction mechanism which eliminates incorrect microtubule attachment. By how? By sensing whether or not centromere is under tension. So by sensing the centromere is whether under tension or not, so the kinetochore corrects the incorrect microtubule attachment. So kinetochore distinguishes between three types of attachments. Let us see one by one. So kinetochore distinguishes between three types of attachment. So it determines which one is correct and which one is incorrect. The first one is amphitelic attachment. Second one is monotelic attachment. Third one is syntelic attachment. So what are these now? Attachment, I'm saying it is an attachment. So attachment means the attachment between the kinetochore and the spindle apparatus of the pole. So to explain the difference between amphitelic, monotelic and syntelic, I'll draw a diagram. See, there are sister chromatids here, to which there is a kinetochore which is present at the close proximity to the centromere, and there is spindle apparatus, spindle fibers at the opposite poles, and kinetochore attaches to the spindle poles or the spindle apparatus, and which helps the attachment of the sister chromatids and anchor to the microtubule. So amphitelic is such type of attachment where the kinetochores are pulled to opposite direction equally. So there is equal force or equal tension built at the centromere in order to pull the sister chromatids towards two opposite poles. Remember, amphitelic means two opposite poles. So is this attachment correct? Yes, this attachment is perfect. So kinetochore distinguishes this correct amphitelic attachment where sister kinetochores are anchored to microtubules from opposite poles. So next type of attachment is monotelic. The word mono means one. Okay, monotelic attachment is where we have, I'll draw the sister chromatids and I'm drawing the kinetochore, and monotelic is where only one kinetochore is attached to the spindle apparatus. So this is monotelic. So this is only where the only one kinetochore is anchored to the microtubule. Is this attachment correct? No, it is not correct. So this eliminates, the kinetochore eliminates this erroneous monotelic attachment. Okay, the last attachment is syntelic attachment. Syn means same. Okay, in this I'll draw the sister chromatids, I'll draw the kinetochore, and I'll draw spindle apparatus. So here in this syntelic, both kinetochores are anchored to the same microtubule which is emanating from the same pole. Is this attachment correct? No. So this may cause aneuploidy, and this may cause the absence of sister chromatids in the daughter cells. There is unequal distribution of the sister chromatids in the daughter cells. So this is distinguished and this is corrected by kinetochore. If there is correction possible, kinetochore does. If does not, so then it halts the cell cycle there itself. So these are the three types of attachments which kinetochore distinguishes by error correction mechanism mediated by Aurora B kinase. The mechanism that monitors and responds to kinetochore microtubule attachment is spindle assembly checkpoint. So spindle assembly checkpoint. This monitors the kinetochore microtubule attachment. So this spindle assembly checkpoint is made up of, it is a complex made up of several proteins which are conserved from yeast to mammals. So the core components include MAD1, MAD2, MAD3 and BUB1, BUB3. So these are the components. These are the components of spindle assembly checkpoint. So SAC, we can say SAC is made up of MAD1, MAD2, MAD3, BUB1, BUB3. So what is this MAD? MAD stands for mitotic arrest deficient. Mitotic arrest deficient. BUB stands for budding uninhibited by benzimidazole. So MAD1, MAD2, MAD3, BUB1 and BUB3. MAD stands for mitotic arrest deficient. BUB stands for budding uninhibited by benzimidazole. All these components forms SAC, which monitors the and responds to kinetochore microtubule attachment during the spindle assembly. So when does they occur? When does they function? So during metaphase. During metaphase, the MAD1 and BUB1 inhibits. So MAD1 and BUB1, not inhibits, they monitor the proper attachment. They monitor proper attachment of what? Proper attachment of microtubule with kinetochore. Microtubule and spindle microtubule and kinetochore attachment. Until proper attachment are formed, until the proper attachment is established, till then SAC inhibits APC-C activity. So if the APC-C activity, if it is not inhibited, so APC-C will initiate the metaphase anaphase transition, you know that. So if that is initiated, then it will cause abnormality. So for that reason SAC inhibits APC-C for a course of time, so that the proper attachment of spindle microtubule to the kinetochore is established correctly. So when problems are sensed, if there is any problem, SAC is activated. SAC is activated. If there is any problem, SAC is activated. I'll write if there is a so there is a problem now. So SAC activated. What happens if SAC is activated? So MAD2, this MAD2 interacts with MAD1 at kinetochore. So it interacts, so MAD2 interacts with MAD1 at the kinetochore, facilitating conformational changes. So this causes conformational changes in MAD2 to promote the formation of mitotic checkpoint complex. So after conformational changes is happening in the MAD2, that promotes the formation of MCC. What is MCC? Mitotic mitotic checkpoint complex. Mitotic checkpoint complex. So if there is a problem, so the problem is sensed, SAC is activated, MAD2 will interact with MAD1 at the kinetochore, facilitating the conformational change in MAD2 to promote the formation of mitotic checkpoint complex. So what is this mitotic checkpoint complex? So this mitotic checkpoint complex MCC is promoted, and the formation is promoted by MAD2 with MAD3 BUB3 CDC20. So it forms a mitotic checkpoint complex with MAD3 BUB3 CDC20. So MCC is a complex of MCC is a complex of what? It is a complex of mitotic spindle assembly checkpoint proteins like MAD2, MAD3, BUB3 and APC-BARC coactivator CDC20. So this is a MCC. So what all components present in the MCC? MAD3, MAD2, BUB3, CDC20. If there is a formation of MCC, so APC-BARC is inactivated, is inactive. Okay. So the MAD2 in the MCC prevents the CDC from binding with APC-BARC. So this MAD2 associates with CDC20, makes it busy, so that it cannot bind and activate the APC-BARC. Why APC-BARC is inactive here? Why it is made inactive? Because there was a problem sensed. So to solve that problem, APC-BARC is inactive with the help of SAC activation. Now you got to know the point. So once kinetochore have established the amphitelic attachment, that is the correct attachment. So now correction is made. Where the stable amphitelic attachment to the spindle is done. So now SAC is silenced. And CDC20 is free. That is, in the sense, MAD2 is not binding with the CDC20. This CDC20 goes and binds to the APC-BARC, making it active. You know the next story. What happens if the APC-BARC is active? So APC-BARC, once it is activated, anaphase promoting complex, once it is activated, so it will start the metaphase anaphase transition. However, merotelic attachment, that is, a single kinetochore is attached to the microtubule from both poles, can go uncorrected as they do not activate the SAC, which leads to segregation error. Okay. So this was about the spindle assembly checkpoint. Next, the third checkpoint is DNA damage checkpoint. DNA damage checkpoint involves signal transduction pathway induced by DNA damage. So it makes sure there is no DNA damage. So it involves a signal transduction pathway. When does this pathway starts? When the DNA damage is sensed, then the signal transduction pathway is induced. Okay, and that blocks the cell cycle progression until DNA is properly repaired. Its function is to ensure incomplete, damaged chromosomes are not replicated. It makes sure these incomplete, defective chromosomes should not undergo replication and passed on to the daughter cells according to the cell cycle stages. So there are several DNA damage checkpoints. So DNA damage checkpoints are classified into three checkpoints, that is G1S DNA damage checkpoint and intra-S phase DNA damage checkpoint, and the last one is G2M phase DNA damage checkpoint. So G1S DNA damage checkpoint inhibits the G1S phase transition in cells that have not yet committed to DNA replication. So DNA replication starts at the late S phase or at the S phase, we can say totally. So that inhibits, so G1S DNA damage checkpoint inhibits the G1S phase transition in cells. So where there is not yet committed, they are not yet committed to the DNA replication. Intra-S phase checkpoint prevents initiation of DNA replication. So to write in short, G1S checkpoint inhibits G1S phase transition in cells which is not yet in cells that have not yet committed to the DNA replication. So what about the second one, right? Intra-S phase checkpoint. This prevents initiation of DNA replication at origin that have not yet been activated. So it prevents initiation of DNA replication at origin that have not yet been activated. Then the last one is G2M checkpoint, DNA damage checkpoint. It inhibits entry into mitosis. So these are the three major DNA damage checkpoints based on the cell cycle stages. The replication checkpoint. So there is another specialized checkpoint, the replication checkpoint. So this replication checkpoint is a specialized branch of DNA damage checkpoint which monitors fork problem. They triggers cellular responses aimed at preserving the genomic integrity. Preserve genome integrity. Okay, so the replication checkpoint is a specialized branch of DNA damage checkpoint, monitors fork problem and triggers a cellular response aimed at preserving the genome integrity. Let's see one by one now. See, cell cycle checkpoint functions to ensure that incomplete or damaged chromosomes are not replicated and passed on to daughter cells. So highly regulated cell cycle depends on series of events mediated by number of checkpoints. So major checkpoints are cell cycle checkpoint, DNA damage checkpoint, spindle assembly checkpoint. Cell cycle checkpoint ensures fidelity of the cell division. DNA damage checkpoint ensures that incomplete or damaged chromosomes are not replicated and passed on to the daughter cells. Spindle assembly checkpoint ensures and guarantees that anaphase onset is initiated only when all chromosomes are properly attached to microtubule aligned at the metaphase plate. See here you can see G1 CDK, G1S CDK, SCDK, MCDK. All these CDK's activities regulated by cell cycle checkpoint. Cell cycle checkpoint verify whether the process at each phase of the cell cycle have been completed, and then it will ensure the accurate completion of the previous phase before progressing into the next phase. Checkpoints in G1 phase are called start in yeast, restriction point in mammals. It is a point at which cell becomes committed to DNA replication and completes a cell cycle. It is during G1 phase that the cell integrates mitogenic and growth inhibitory signals and makes the decision to proceed, pause, or exit the cell cycle. So this was about the cell cycle checkpoints. In our next class, we'll take up another concept, that is DNA damage response signaling pathway, and we'll discuss about role of RB protein. What is this RB protein, and what its role in cell cycle regulation? That will be dealt in the next class. So see you. Thank you.